Cells are the basic unit of all living things. Most of the cells have three common parts: cell membrane, nucleus, and cytoplasm. This is the nucleus. It is the most conspicuous organelle found in an eukaryotic cell. The nucleus is separated from the cytoplasm by a membrane called the nuclear envelope. The nuclear envelope is a double-layered membrane, and it is perforated with tiny holes known as nuclear pores. This dark spherical structure is nucleolus. It is present in a fluid-like medium known as the nucleoplasm. So nucleoplasm is the fluid contained within the nucleus in which the chromatin material and nucleus are present. Chromatin material condenses into a highly ordered structure called chromosomes. Chromosomes carry genes that are units of heredity information. The cytoplasm consists of all of the contents outside of the nucleus and enclosed within the cell membrane of a cell. It is composed mainly of water and comprises of various organelles floating in it. A simple-looking cell membrane is actually a very complex structure. The cell membrane is primarily composed of a mix of proteins and lipids. Phospholipids are a major component of cell membrane. They form a lipid bilayer that has proteins, cholesterol, and polysaccharides embedded in it. Structural proteins help to give the cell support and shape. Transport proteins are embedded in the cell membrane and help the cell in transport of substances across the membrane. The cell membrane controls what moves in and out of the cell. The membrane is selectively permeable. It permits entry and exit of only some substances in the cell. That's why it is known as selectively permeable. The movement of substances across the membrane usually takes place through diffusion. Diffusion. It is basically a spreading out process. Diffusion allows the movement of particles from an area of high concentration to an area of low concentration. For example, if you drop some ink in a beaker of water, the ink particles would end up spreading out, eventually becoming evenly spread out throughout the container of water. After this point, the ink particles would continue to move, spreading to get evenly distributed throughout the water. Plants also exchange the gases through diffusion. The gases diffuse into the intercellular spaces of the leaf through stomata, which are present in epidermal cells. Once the molecules diffuse, they move to the cells that require them. Facilitated diffusion. The molecules, which are not lipid soluble, are transported with the help of some transport proteins. This type of transport is known as facilitated diffusion. During facilitated diffusion, transport of the molecules is carried out with the help of some carrier proteins. During facilitated transport, carrier proteins assist in moving these large molecules across the cell membrane down its concentration gradient. Like diffusion, no energy is required to transfer the molecules during facilitated diffusion. Sometime, even the carrier protein changes its shape temporarily to deposit the molecule on the other side. There are certain proteins which are globular in shape and have pores that form a sort of channel which help transport molecules in and out of the cell. Diffusion and facilitated diffusion both are passive type of transport and do not require any expenditure of chemical energy. Simple diffusion and facilitated diffusion are considered as passive kind of transport, as no energy is required during transfer of substances across the membrane. There is another type of transport in which energy is used. Such transport is known as active transport. If passive transport is like driving your cycle down a hill, then active transport is like using energy to pedal your cycle up a hill. Now let us see how active transport takes place. During active transport, substances are moved against their concentration gradient using some energy. During active transport, a carrier protein acts as a pump that causes a substance to move against its concentration gradient. The sodium-potassium pump carries sodium ions to the outside of the cell and potassium ions to the inside of the cell. Energy in the form of ATP molecules is required for active transport to occur. When a cell is dipped in any solution, the volume of net movement of water in and out of cell depends on the solution's concentration in which it has been kept: isotonic, hypertonic, or hypertonic. Hypotonic solution. When the concentration of dissolved solutes in a solution is less than that of a cell, it is known as hypotonic solution. Animal cell, when kept in hypotonic solution, swells up due to inflow of water. It becomes bloated and at times bursts and dies. When plants are placed in water, the water enters the plants through osmosis and fills up the cell to its maximum capacity. A strong cell wall stops the cells from bursting and provides turgidity to the cell. Isotonic solution. When the concentration of dissolved solutes in extracellular fluid is the same that of a cell, it is known as isotonic solution. When the concentration of dissolved solutes is the same that is inside as well as outside of the cell is said to be in an isotonic environment. This creates a dynamic equilibrium that maintains the status of the cell. Hence, no change takes place in the shape of the cell. Hypertonic solution. When the concentration of dissolved solutes in extracellular solution is less than that of a cell, it is known as hypertonic solution. Animal cell, when kept in hypertonic solution, results in crenation, where the shape of the cell becomes distorted and wrinkled due to the loss of water by the cell. When a plant cell is placed in a hypertonic solution, the water inside the cells is drawn out by osmosis. The vacuoles decrease in size. The cytoplasm also shrinks away from the cellulose cell wall, and the state of the cell is known as plasmolysed cell. This causes wilting in the plant. An egg is always a single cell. For performing osmosis in an egg, we need shell-less eggs. A shell-less egg will represent a cell surrounded by a selectively permeable membrane. The shell of an egg can be removed by soaking the egg in vinegar, as it is made up of the mineral calcium carbonate. As vinegar contains acetic acid, it dissolves eggshell, which is made up of calcium carbonate. During this process, it releases the gas carbon dioxide in the form of bubbles. Actually, calcium carbonate reacts with acetic acid and breaks down into carbon dioxide, water, and calcium acetate. After the shell has been dissolved, only the membrane remains around the egg. Remove the eggs from vinegar and rinse them gently under the tap water. Weigh both the eggs. Egg A weighs 98 grams and egg B weighs 102 grams. Put egg A in hypertonic solution, say water. Dip the egg B in hypertonic solution. It can be thick sugar solution or salt solution in water. Leave overnight. Rinse and weigh again. Egg A is now of 104 grams, as egg A was present in hypotonic solution. Water has entered the cell, thus increasing the mass. Egg B weighs lesser than previous weight. Now it weighs only 62 grams. As this was present in hypertonic solution, water from the cell was lost to outside. Due to the loss of water by the cell, weight decreased drastically